about factors protecting total nucleated cells in the umbilical cord blood units. Okay. So first we'll be talking about the umbilical cord blood in general, then our research in particular. Then we'll talk about the methodology, we'll discuss the results, and then our conclusion. So in 1988, that was the first successful use of umbilical cord blood in the hematopoietic stem cell transplantation. It was done in France on a patient who was suffering from Fanconi anemia. And since then, it was being used as an alternative source of stem cells. It has the advantage of having less rigid requirement for tissue matching, the HLA, and fewer complications, mainly the graft versus host disease. Also, fewer chances of infection transmission. So, in general, the umbilical cord blood transplant is potentially curative. We have hematological disease, whether malignant or non-malignant, metabolic, and even immune diseases. So, since then, thousands of transplants have been done around the world. And because of this recent expansion, many private and public banks were set up for the storage of the units. However, the cost of having these banks is not insignificant. The cost of single units is around $3,800 US. This is involves collecting, processing, and storage. However, up to 60% reported that 60% of these units are discarded, even before doing the transplant. Why is that? Because of the low cell content, which is the major drawback of the umbilical cord blood. So it makes it an issue of cost effectiveness, especially for the public and for the non-profit banks. So many studies that shows that the efficacy of the graft, survival of the patient, depends mainly on the TNC, which is the infused total nucleated cells, and also the CD34. So the number of these two. Also, there is recent, also the colony forming units. There is a study published in 2011. It says that colony forming units are now the major independent factor or parameter predicting the survival of the unit. However, still many banks use the TNC count. CD43 has many issues with how the labs process it. So many efforts are concentrated on the TNC count. So our research recently in the Middle East, especially in Saudi Arabia, there are many public and non-profit banks who are set up for the donation and storage. So we try to adapt we try to look in the literature review to see what factors can predict the success of the graft. However, there are many variations exist, so we try to look into our own data and look if there's factors that are special in our culture. Our next object was to compare the outcomes internationally and look whether these factors are really comparable to other results. So our aim was to optimize the clinical use of the umbilical cord blood hematopoietic stem cells and improve the criteria of donor selection. We tried to look into 17 variables, whether it's related to the mother or the infant. We wanted to know what is the percentage of the clinically accepted cord blood units and how many are rejected. We want to know the factors that can influence the TNC count. So this is cross-sectional studies. It was done in our hospital in Riyadh. So the explanation to mothers and consent was obtained in the outpatient clinic, and then the blood collection was in the labor and delivery, and then the storage was in the cord blood bank. We calculated our sample size to be 957, and we did a convenient and consecutive enroll technique. However, in the inclusion criteria, we only included medically free mothers and newborns. Cord blood collection, it was only ex utero, meaning after delivery. After delivery, we collected the cord blood. And then for the ethical issues, we obtained the IRB approval and informed consent. We entered into the SPSS, and we set the mean and standard error and frequency. Also, our p-value was less than 0.05. Confidence interval 95%, and we used the ordinal logistic regression. So these were the variables that we used. For the mother's age, and then the placenta, gestational age, the maternal blood indices, and then the cord blood, and then we tried to know if the gender is related or not. Then the obstetric variables such as gravity, barity. Also, we tried to look into the mode of delivery. So we based our results and this accepted level of banking, which is used internationally, and based on it, we divided our units into 3 categories. The first one, which is thankfully the majority, with 55.6% being clinically accepted. However, only 19% was totally discarded. The rest was for only research use. So our results, which is about 50% was rejected. It's compatible with the results international. Whether the 17 variables that we discussed before were clinically significant or not, only 3 factors that appears to have a clinical significance. First is the cord blood volume. Second is the newborn birth weight, and then the mode of delivery. So you can see that the p-value of the cord blood volume predicts that this is the most important one, and then the weight, and then the mode of delivery. However, further result interpretation, the cord blood volume appears to be the best predictive value. However, for every 10 grams increase from the mean cord blood weight, which is 74.22 gram, there's 2.6 increase. It's 2.6% more likely to produce TNC count being clinically accepted. For the birth weight, for every increase, for every 500 increase from the mean, which is about 3 kg, there's a 40% more likely to produce a clinically accepted TNC count. For the mode of delivery, C-section are 40% less likely to produce clinically accepted TNC count. So to compare our results with those that are done internationally, many of these previous results were limited by having small sample and a small number of variables examined. Some of the previous research had in utero cord blood collection. However, it was demonstrated that this in utero method produced less TNC count and also more likely to transmit infection. And other studies have set chronic disease, such as diabetes and hypertension, as variables with different results. So some studies said that being a diabetic mother produces a significantly less a successful graft, while other studies discarded that. So in contrast to our studies, mode of delivery did not show any statistical significance in other studies. However, maternal age, which is mainly mothers less than 20, gestational age with the newborn being term and post-term, parity, especially para, infant sex, male. These factors show statistical significance in other studies, which did not show in ours. However, the majority of the research done before were consistent in that these variables did not show any significant relation to the TNC count, the gravity, abortion, the placenta weight, and then the blood indices of either the maternal or the newborn. So our limitation, so our local lab that does not process either of these parameters. Also, we have minimal racial variation in our country, which limits our research in that area, because some other research showed that ethnicity has a significant role in the TNC count, especially the Caucasian race. So, in conclusion, several maternal and neonatal factors appear to play a major role in predicting accepted TNC count. However, we recommended newly founded cord blood banks to have a local data in order to see what are the significant variables that play a role in their population, so they can optimize sort of blood donor selection, which would be cost effective and also trying to preserve the quality of the cord blood units. Our next step would be recommended other studies of the outcome of our local cord blood banks outcome, and compared to other banks that using the CFU or the CD34. Okay. So I'd like to thank all the participants, the mothers, the staff of the cord blood units, the hospital, and my university. These are my references. Thank you. Yeah. Yeah.
Yeah, that was a very good presentation. I'm impressed you were able to get 900 and something people to participate in it. It's quite a huge.
Yeah, it took a long time to get first to explain to the mothers, and then to get the consent, and then follow up in the labor and delivery, and then they get the actual sample, and then it took a lot of time.
So, yeah, absolutely. Is that right? So all of the 900 plus mothers, this data is for all of them.
Yeah.
Amazing. Right. I was just wondering whether, this, you mentioned that cesarean sections led to about a 40% less likelihood.
Yeah.
Is there any particular reason why you think that might be so?
To be honest, I don't know. I found the same results in other researches. However, some research said that that it may reduces the TNC count, but overall it may not have a significant effect on the success of the graft itself, but there's a significant variation